Welcome, everyone, to this webinar on Translational Allostery from Cryptic Binding Pockets to Latent Networks, brought to you by the Protein Society. It's wonderful to be here together to focus on this really central topic and challenge in protein science. So thank you, everyone, for being here. My name is Liz Meyering. I'm currently serving as past president of the Society, and it's my great pleasure to introduce the webinar today. Before turning things over to our organizer, Giuseppe Melacini, I have just a couple of brief announcements. So I'd like to just alert you to our next webinar, which is actually next week, on another important and exciting topic, Introduction to Protein Design for Africa. And you can still register for the webinar on our website, as you can for all of our webinars. I also want to invite you to join us at the 40th Anniversary Symposium of the Protein Society, which is taking place in July in Boston. We have a stellar program, which you can see at our website, and you can still register to learn about the latest in protein science. And there's also still availability to register for poster presentations in the poster competition. The deadline there is June 1st. So I hope to see many of you there. And I wanted to mention also next year's symposium, which will be July 9th to 12th in San Francisco. So you can put that in your calendar. And I also want to just finish with highlighting our Q1 journal, Protein Science. We have a fantastic editorial team led by John Currian, and the journal publishes papers encompassing a very wide range in outstanding protein science. And I wanted to just note, for those of you who may not know this, that this is a society journal. Most journals are not society journals. So protein science is led by and for protein scientists. And because of this, everything that the journal does and contributes to our protein science community. So one example of this is that we have Best Paper Awards for first authors who are graduate students or postdocs, and they're invited to give a talk at the annual symposium, which the journal also supports, along with many other activities. So I encourage you to think about this for your work. And that brings me now to our main event. I want to warmly thank Giuseppe Melacini for organizing and serving as webinar chair, and all of our speakers for sharing your exciting research with us today. A few words about Giuseppe. He obtained his PhD in biophysical chemistry from University of Milan in collaboration with Murray Goodman at University of California, San Diego, and then did postdoctoral work with Robert Kapteyn and Rolf Bollens at Utrecht University. He joined McMaster then as an assistant professor and is now professor and chair of the Department of Chemistry and Chemical Biology with joint appointment in biochemistry and biomedical sciences. He's an expert in biomolecular NMR and has pioneered methods such as Cheska for defining allostery, as well as dynamics, and focusing on a molecular understanding of proteins involved in Alzheimer's disease, cancer, and cyclic nucleotide signaling pathways. He's received numerous distinctions, such as recently the Canadian Society for Chemistry Biological and Medicinal Chemistry Award and Excellence in Graduate Supervision Award. He's authored over 110 papers and served in leadership roles within the Biophysical Society of Canada and on editorial boards such as JBC, Journal of Biological Chemistry. So before turning this over to Giuseppe, I want to just say some logistical notes. So each speaker will present for 20 to 25 minutes, and then there'll be a brief question period. After the talks, we'll also have further questions in a discussion format. So everyone, please take a moment to find your Q&A tab, where you can type your questions at any time, and only the organizers and speakers will see them. So questions will be read out by our chair, webinar chair, and answered live, or they may be answered also in the chat. So these questions really enrich the discussion, and I want to thank everybody for participating in the webinar and contributing in all these different ways. And that's it for me. Giuseppe, it's over to you.
Thank you so much, Liz. I would like to start saying a few words about the topic of the webinar. We know that the translational potential of protein allostery is now quite well established. We know that allosteric sites are subject to lower evolutionary pressure compared to active or orthosteric sites. So targeting allosteric sites offers an opportunity to increase selectivity and decrease off-target effects. Probably one of the best known examples of the effectiveness of targeting allosteric sites is provided by the allosteric inhibitors of ABL tyrosine kinases, which are actually quite effective against chronic myeloid leukemia, especially when resistance mutations arise in the ATP binding site. But the translational potential of allostery is not limited only to kinase inhibitors. For example, cryptic allosteric pockets accessible only in excited states of proteins have opened very many opportunities to drug targets that in the past were considered completely undruggable. Another example is the central role of allostery in the regulation of systems ranging from CRISPR-Cas9 for gene editing and biomolecular condensates, which are actually redefining our understanding of what a drug target is. Now, these advances have been made possible by the integration of computational methods and experimental techniques, ranging from NMR, MD simulations, and AI modeling. Today, we are very fortunate to have here 4 leaders in these domains: Julie Forman-Kay, Giulia Palermo, George Lisi, and Greg Bowman. They were selected not only because their name starts with G or J, that's actually purely a coincidence. They are here because of their seminal contributions to the field. So today we're going to start with Greg Bowman first, and I would like to introduce Greg. So Greg received his bachelor summa cum laude from Cornell and his PhD from Stanford. After a postdoc at UC Berkeley, he joined the faculty at Washington University, but in 2002, he moved to UPenn, where he's currently an endowed professor, Louis Heyman University Professor of Biochemistry, Biophysics, and Bioengineering. He's also the director of the Folding at Home Distributed Computing Program. Greg is the recipient of many awards and distinctions, but for the sake of time, I'll just cite a few: the 2024 Hirschfelder Lecture at the University of Wisconsin-Madison and the 2023 Graham Lecture at the University of Virginia. The Bowman Lab focuses on developing therapeutics and interpreting genetic variants by understanding and exploiting protein dynamics through a combination of experiments, machine learning, and physics-based simulations. Without further ado, I welcome Dr. Bowman, Greg, to present his talk on cracking undruggable proteins with cryptic pockets. Take it away, Greg.
Thanks so much. You can hear me, right, Giuseppe? All right,
yes,
good.
Yes, sir.
Perfect, thank you. So yeah, it's a pleasure to be here. Thanks, Giuseppe and the Protein Society for the invitation and for putting this all on. As was mentioned, my lab, we're really obsessed with all things protein dynamics, but particularly in cryptic pockets and allostery, right? And so just to motivate this, the idea, if we back up and think about the current state of rational drug design, right, is that the best case scenario is that you have some enzyme or receptor that you're interested in inhibiting, right? And if you were to, and hopefully you have a structure of your protein and you have a nice deep pocket at the key functional site that you want to inhibit, right? And so by using the structural information from your structure and your understanding of physics and chemistry, you can hopefully design a small molecule that will bind to that pocket and form lots of energetically favorable interactions in every direction to give you the high affinity and specificity that you want for a drug, right? And so this is great when it works out. We've all benefited from this, but unfortunately, it doesn't always work out, right? Oftentimes, things go awry very early on because you say, hey, I have this biologically validated target from Ebola virus that I want to inhibit, and someone solved the structure, so I'm going to use that to design an inhibitor. I know I need to target this functional site around this red residue, and now you're already in trouble, right? Because there's no nice deep pocket here, right? This is a super flat interface compared to over here. And it's really hard to get high affinity interaction with a small molecule when basically only one face of the molecule is going to be able to interact with the protein instead of every possible surface on the molecule. And so based on very simple criteria, like, is there a nice concave pocket that a drug-sized molecule can wedge into? It's estimated that only some small fraction of our proteome is druggable, right? And this is a big problem for many of my colleagues at the medical school who spend all this time finding the critical targets for various diseases. And then they come and they say, hey, Greg, can you help us? And I say, nope, nothing to do. Go find another one, I guess, right? But fortunately, everyone here is probably familiar with protein dynamics and the fact that, yes, it could be that in the ground state of your protein, there's no nice deep pockets, but there's all these excited states, and those could harbor unexpected or cryptic pockets that would allow us to target these proteins that one might consider undruggable if we over-focused on the structure of the ground state, right? And this could open up all kinds of cool opportunities. So we could take proteins that appear to be undruggable based on extant structures and realize that there's actually a way to drug them. As Giuseppe mentioned, we could get specificity in difficult cases. And one of my favorite things to noodle on sometimes is like, could we use cryptic pockets and allostery to actually enhance desirable activities, say to like counter the effects of some deleterious mutation? Right? So all kinds of potential uses for this. So we've done a lot in the past with developing simulation algorithms for taking, undruggable proteins like this, looking for, moving parts that might give rise to cryptic pockets. So here, hopefully you're seeing this nice large concavity open up that exposes these orange residues that are buried in all the available crystal structures, but get exposed to solvent and drug-sized molecules by the opening of this pocket, right? So we can find these pockets. We have all kinds of ways of quantitatively teasing out allostery from these simulations. And now half of my lab is experimental, right? So we have a whole battery of tests for, is there a pocket? Is there allostery that can control function, and so on, right? So for today, I'm not going to talk about that. What I'm going to back up and say, like, well, I've had this dream since I began my lab, right? Which is that, these simulations are expensive, takes a bunch of time. There's all these things that we could go after, right? And so it'd be really nice if we had an algorithm where you could take one structure of a protein, say the crystal structure, run this algorithm in fractions of a second and get back a prediction of, if and where cryptic pockets are likely to form, and ideally, whether they would be functionally relevant, right? So now we're post AlphaFold, right? This is something I started scheming about well before AlphaFold. And so you might think, great, we have all this data in the protein data bank, right? We can go AlphaFold upon it, or some related thing, and we can predict cryptic pockets, right? But it turns out, despite the huge number of structures in the protein data bank, there's only like 100 examples where we have, the apo or unbound structure where a pocket is closed, and then a structure where something is stuck in the pocket holding it open, right? So that's not nearly as much data as we'd like to have or need in order to do a purely structure-based, machine learning approach. And so as things have gone on, what we realized is like, wow, our lab has worked on a whole lot of different proteins right now. This is just a sampling that shows everything from our little 120 residue Ebola protein that we've done a lot with to the 3,600 residue spike protein from coronaviruses, right? So we have a whole lot of diversity and structure and sequence and a lot of data. So at this point, in the last like 10 years, we have generated tenths of a second of simulation, right? So if we say, just to put it in perspective, that a typical good simulation paper will have like 1 microsecond of simulation data, right? People are starting to surpass that on a more regular basis, but we'll say that, right? Then a tenth of a second is 1,000,000 times more data than a single paper, right? So we've got at least 1 million, a couple million-fold times more data. And anytime you see a big pile of data, an exciting question to ask is like, well, what could we get from throwing machine learning at this, right? So what we did a couple of years ago is develop this algorithm called Pocket Miner that uses a graph neural network to predict cryptic pocket formation, right? So what it does in the end is we can give the algorithm an input structure, right? It's going to make a graph representation of this and use a message passing algorithm to predict what's the probability that each residue in this protein participates in a cryptic pocket. And one of the cool things about this is the way we trained it was on simulation data. So we take short snippets of simulation, like 40 to 50 nanoseconds, right? We label residues as having participated in a cryptic pocket or not. And that just means, was it buried in the starting structure for that snippet of simulation? And did it get exposed to solvent, suggesting it's part of a cryptic pocket or not, right? So those are our labels. And now we're going to train the network, given the starting structure, predict those labels, right? And it turns out that by training the algorithm in that way, now we can go and hand it, crystal structures or alpha fold structures or cryo-EM structures or whatever source of information, structural information you got. And it can do a really good job of predicting which residues are most likely to participate in a cryptic pocket. So I won't go through the quantification of the performance. I just jump to like one of the cool things we can do with this now, because it takes, something in milliseconds, fractions of a second to run on a specific input structures. We can start running it on large sets of proteins, like 10,000 proteins that are implicated in cancer, right? And so prior to our work, people had said in this particular group of 10,000 proteins that only about half of them were potentially druggable because of simple criteria, like, is there a pocket at the functional site that you want to inhibit? And what we were able to come and do is say, actually, these 50% of the proteins that people had thought were undruggable, more than half of those look like they have nice cryptic pockets. And I think this is a conservative estimate, and the fraction is probably much higher, right? And so now we can start thinking about things like, well, which of these would be, which of these cryptic pockets do we have the most confidence in, for example, or which targets that are most important for cancer, have cryptic pockets that I'd like to go for. So this opens up all kinds of opportunities. All right, so this is really great. Still challenges though, right? So one of the things, coming to the theme of allostery, is that these graph neural networks are intrinsically focused on local information, right? So when we make a prediction in Pocket Miner for a given residue, we're just building a graph that knows about the 30 nearest neighbors to that residue, right? And it doesn't know explicitly anything beyond that. The representation for each residue in the protein gets updated a few times, right? So there's some implicit information about residues that are further away. But because of the way messages are passed around, that's going to fall off as a function of distance. And you might not pick up on these really cool long range effects that could be important. And so what we did is say, oh, well, maybe we could do better if we borrow the idea of an attention mechanism, right? So now, in addition to having, our graph-based neural network that's got this very, focused on local information, now we're going to add a layer that connects every residue to every other residue, potentially, right? One of the things here you could imagine is like learning, strength of interaction between every pair of residues, right? It's a lot of parameters. So one of the cool things with the attention mechanism is really learning, like, which of the other residues are important, given what I've picked up on from my training data, right? So it really focuses on finding what's important. And so this gives a way to get information over long ranges and also to tell, well, which residues are important for determining, my cryptic pocket formation. So in this case, we have this cryptic pocket in this red region. That's the high probability region that's pocket. And then using the attention scores, we can also say, hey, these residues and spheres are having a large outsized influence on the probability that this pocket in red is going to open or not. So those might be places that I want to consider making mutations to control pocket opening, or parts of the protein that are more likely to be impacted if I were to find some way of targeting this cryptic pocket. So this was really fun. The attention stuff has improved performance, right? Because there actually is long-range stuff going on. There is allostery. So having this allows us to do better at predicting the probabilities that a residue participates in a cryptic pocket. And we also get this level of insight into allostery and can start saying, not just is there a pocket, but is targeting that pocket likely or not to have some control over the function of my target. So there's some cool tricks here I won't have time to go into, right? When we add this, we add the attention mechanism, we add a bunch of parameters. So we also needed more training data. So we did this in an active learning style. So we asked what proteins is Pocket Miner doing worse on? And then we went and got simulation data for those and added it into the training set. And this worked out really nicely. So just to give one example where we've taken it all the way to experiments is we ran this on all the human proteins in the PDB with like less than 300 residues that have buried cysteines, less than 3 buried cysteines, but buried cysteines that look like they're part of a cryptic pocket, right? Or that have buried cysteines. And then we can assess how likely those cysteines are to participate in a cryptic pocket. And this is really nice because we have assays for measuring the accessibility of cysteine, so we can test if a buried cysteine is actually getting exposed to solvent by the opening of a pocket. So this gave us 10,000 candidates still, which was a bit overwhelming. So we kind of arbitrarily said, well, I don't want Alzheimer's, and it'd be nice if we could do something about that. So of those 10,000 proteins, are any implicated in Alzheimer's? And this led us to an enzyme called delta secretase that is responsible for cleaving A beta out of its parent protein, right? So this is the structure with color-coded things. Red is high probability that each residue participates in the cryptic pocket. I've highlighted the 3 native cysteines. So one is an active site residue, and then there's these 2 other cysteines, right, that are buried but have some reasonable probability of being part of a cryptic pocket. And if we now look at attention scores of residues, like how much attention when I make the prediction for the active site does every residue have, this cysteine over here lights up bright red, right? So this is a buried cysteine that high probability, reasonably high probability of being part of a cryptic pocket, and reasonably high probability of being allosterically coupled to the active site, right? So this is great, suggests we found something, potentially interesting and new. So now what we can do is start running simulations. So we can, apply our pocket hunting simulation algorithms. And what we find is that sometimes, the active site is open. So a cyan blob is a space-filling representation of the active site. But sometimes the active site closes up and there's this pocket this blob over here next to our cryptic cysteine 219, right? And so there's this trade-off between these, which again suggests that if you found something that targeted this cryptic pocket, you would close up the active site and inhibit this enzyme, right? So now we can turn to our thiol labeling assay. So we're introducing a chemical labeling reagent called DTNB that can react with exposed cysteines. What you can see is we see 3 cysteines being labeled. We have 3 observed rates at each of our DTNB concentrations. And importantly, all of those rates are well above this dotted line, which is the labeling rate that we would expect based on global unfolding of the protein, right? So what that tells us is that our active site cysteine is exposed, but sometimes it's buried, right? Because otherwise the labeling rate would be much faster. And both of our buried cysteines that we predicted are part of cryptic pockets, including the C219. So that's great. We have a pocket as predicted, and we lucked out. And digging through the literature, it turns out that people have already shown that if you mutate that cysteine, you inhibit the enzyme, right? So we also know from this extant data that there's allostery between these sites. So this was a really nice confirmation of the predictive power of the algorithm in a setting where no one had talked about a cryptic pocket before. So we've got a couple other examples that I'll skip over for the sake of time. One that's really interesting is that all of our training data, essentially, except for one protein, is a globular soluble protein. but the algorithm works on GPCRs like CB1. And we've also done a lot with this Ebola protein, showing that we can predict residues where mutations either enhance or suppress the probability of pocket opening over here, right? So this is really useful for ranking potential targets and also for designing mutants to control pocket opening that can be useful tools for downstream assays. All right, so with that, I'll thank my group, all the people around the world that have helped run simulations through Folding at Home on our funding sources. And again, thanks for the chance to talk and for all of you for listening.
Thank you, Greg, for an excellent presentation. We already have a couple of questions. The first one is about the druggability of cryptic pockets. So once you find the cryptic pocket, can Pocket Miner also predict the druggability of that cryptic pocket?
You know, no, I don't think so, right? And you know, my opinion right now is that druggability is a pretty ill-defined term in any sort of quantitative way, right? So we do have some surrogates though, right? So one of the things that we'll do is take open structures from our simulations and run them through the tools like FTMap to get a sense of like, are there multiple drug fragment probes that form favorable interactions with different parts of the pocket? So I think that at least gives a qualitative answer, but I don't think there's a good quantitative definition that we could really train Pocket Miner on.
Okay, thank you, Greg. We also have another question about whether your pipeline, I guess, meaning Pocket Miner in its different versions, have been tested on membrane proteins like GPCRs, for example.
Yeah, so we just posted this preprint this week, but we included work on GPCR called CB1 that's of interest as a target for circumventing the opioid epidemic, right? And so there's a known cryptic pocket that was recently published that's up here. So all these residues I've highlighted in spheres are residues in a structure of a ligand bound in the cryptic pocket that line that cryptic pocket. And you can see that Pocket Miner has labeled them all bright red, right? So we've successfully recapitulated that pocket and then have a prediction that maybe there's something else going on down here also, right? But this is a nice first glimpse that this can work on membrane proteins as well.
Thank you so much, Greg. And there are several other questions coming in the Q&A, but in the interest of time, I would suggest that we move on to the next speaker, Giulia Forman-Kay, and we'll circle back to the questions towards the end of the webinar when we have the discussion time. Thank you again, Greg.
Pleasure.
So it's my pleasure to introduce Giulia Forman-Kay. She received her Bachelor in Science in Chemistry from MIT and her PhD in Molecular Biophysics and Biochemistry from Yale University, I guess more or less at the same time that Louis Kay and David Wishart were there. So those were magical years at Yale. After a postdoc at NIH with Marius Clore and Angela Gronenborg, she joined the Hospital for Sick Children here in Toronto and the Department of Biochemistry at the University of Toronto, where she's now a Canada Research Chair in Intrinsically Disordered Proteins. Since 2015, she has been serving as program head of the Molecular Medicine program at Sick Kids, and as well as founder and co-director of the Structural Biophysical Core at Sick Kids. Dr. Forman-Kay is the recipient of many awards and distinctions, but just to cite a few, in 2016, she was elected Fellow of the Royal Society of Canada. In 2021, Fellow to the Royal Society of London. She's also a Fellow of the Biophysical Society of Canada, which is the highest honor conferred by that society. The Forman-Kay Lab is a leader in the biophysical chemistry of IDPs and liquid-liquid phase separation, and studied primarily by NMR and computational approaches, and has made many seminal contributions on how dynamics relate to function. Without further ado, I welcome Dr. Forman-Kay to present her talk on biomolecular condensate mechanisms of allostery. Take it away, Giulia.
Okay, thanks so much, Giuseppe, for the invitation and to the Protein Society. So just to correct, I'm no longer molecular medicine program head since November, but thank you for that lovely introduction. So I'm just going to start by pointing out that intrinsically disordered proteins and protein regions that lack stable secondary and tertiary structure make up a third of the residues in the canonical human proteome as defined by state-of-the-art prediction tools such as spot disorder. What's very interesting, Reed Alderson found a number of years ago, is that if you look at the residues that are predicted to be disordered and you compare them to the residues that AlphaFold 2 gives a confident prediction of structure for, you see that 15% of the highly predicted disordered residues have confident AlphaFold 2 prediction with these PLDT scores greater than 70. And this is due to AlphaFold 2's use of deep multiple sequence alignments, and positional conservation is diagnostic of structure. So what this is not saying that the disorder prediction is incorrect. It's that these residues are conditional folders so that they fold upon binding or post-translational modification. So AlphaFold 2's structure for the rest of those residues that aren't confidently predicted are obviously not meaningful, but the structures for conditionally folded states are not always accurate either, because different binding partners or PTMs fold IDRs into different structures. So it convolutes the information in the multiple sequence alignments. But that is not the focus of what I wanted to pull out of this study. The main excitement for me here was that a majority of disordered protein residues in the human proteome likely never acquire structure, even transiently. So, there's only a small fraction that are conditionally folded. So what do these IDRs that don't conditionally fold do? And I like to think that they enable regulatable, because they are the major sites for post-translational modifications, regulatable dynamic interactions with all sorts of other biomolecules. And these include in discrete complexes and in large scale associated states. So discrete complexes, I'm just showing this example of the cyclin-dependent kinase inhibitor SYK1 and its interaction with a component of a ubiquitin ligase, CDC4, that enables a sharp cell cycle switch in yeast. It involves 6 or more phosphorylated elements that bind of SYK1 that bind to either the primary binding site on CDC4 or a negative allosteric site. And the binding to the negative allosteric site kicks the bound state off of the primary site, just enabling this dynamic exchange, which allows the system to be able to count to 6 and give a very sharp cell cycle transition. But what I want to focus on for the rest of the talk is the large scale associated states. For example, in phase separated conditions, and phase separation being starting from a mixed phase where you have solutes dissolved in solvent, and then you demix into solute enriched solvent depleted phase and a solute depleted solvent enriched phase. And these phase transitions that involve this demixing process are key for the formation of biomolecular condensates, which can be described as non-membrane encapsulated compartments in biology. These include things like the nucleolus in the nucleus, and P-bodies and stress granules in the cytoplasm. And Michele Vendruscolo and Magna Fuchs writers' work with fuzz drop predicted a number of years ago that more than 80% of human proteins are found within condensates at some point. So these condensates are driven by multivalent dynamic interactions involving folded binding domains for either IDRs, motifs within IDRs, nucleic acids, or other folded domains. Of most interest to me are the dynamic interactions of chemical groups within IDRs for other chemical groups or for nucleic acids, and nucleic acids themselves can phase separate. The condensates that are formed can range in size from below 10 nanometers to over 5 microns, like nucleoli. And since small condensates are below the detection of light microscopes, you might say, well, how can you define what's a condensate? And so my definition of a condensate is something that enables distinct inside and outside solvent environments, with the inside solvent having significant contribution from protein and nucleic acid. So condensates can have hundreds of components, but we can make a simplified categorization of these components into scaffolds, which drive condensation and create the unique solvent, and the clients, which partition into the condensate and act as solutes. And so if we think about in an in vitro condensate experiment, for instance, the dilute phase, water is the solvent, and in the condensed phase, proteins and nucleic acids also form a significant component of the solvent. And what that means is you go from, in the dilute phase, from a high dielectric constant to a lower dielectric constant, and that can then control the energetics of protein stability, binding, and other conformational equilibria. So what do condensates do? Many things. They make materials, extracellular matrix, the nuclear pore, they nucleate cytoskeleton. But of most interest to me is they make compartments with unique solvents that control biochemical reactions. And today's webinar is about allostery. We all understand that enzymatic reactions are often regulated by allostery. So how is allostery related to condensates? So we can think about condensates being within a condensate or not, being an allosteric switch, because the environment of being inside a condensate could alter allosteric equilibria. And I'm going to spend most of today's talk talking about this. We can also think that condensates could just in general be a crucible for allostery, because very high concentrations are going to lower the barriers for allosteric interactions, for instance, with allosteric ligands. Alternatively, condensates themselves can form or dissolve as the result of an allosteric switch. And there was a very interesting recent paper from the Sao group that DNA binding releases a disordered region to trigger an allosteric switch for phase separation of the heat shock transcription factor 1. So binding or post-translational modifications that we think of like as allosteric switches can change the structure or dynamics to prevent or promote phase separation. But going back to that first focus on sort of changing the environment, we need to consider that the definition of allostery has changed over the years. So traditionally, it was understood as a perturbation, binding, or post-translational modification at a distal site to impact an enzyme active site or a protein-protein interaction site via a discrete cascade of structural changes that linked the two sites. But then, and now currently understood, is there's a understanding of the ensemble nature of both folded and disordered protein states. So allostery can be thought of as a distal perturbation that changes the energy landscape with structural shifts, so shifts in the populations of states, or dynamic shifts where you're, let's say, narrowing this well that ultimately impact the active sites or binding sites. And so what I'm somewhat hesitatingly proposing today is to stretch this definition to multivalent perturbations, where you have dynamic, multiple dynamic weak solvent-like interactions in the context of a biomolecular condensate that then can change the energy landscape, and that we can consider this also allostery. So with that in mind, I want to tell you a story of how condensate solvent interactions impact protein conformational equilibria to demonstrate this allosteric consequence. And it's a work that was done by a superb postdoc, Rashik Ahmed, who's jointly supervised by myself and Louis Kay, and also in very close collaboration with Louis Kay. So it's very hard to think about the conformational states of proteins inside condensates because it's difficult to experimentally examine them. They're too dynamic for cryo-EM and X-ray crystallography, and therefore we need to use NMR tools. There's a dynamic range issue because we're talking about clients that can be 100 to 10,000, 100 to 1,000 fold lower in concentration than the scaffold. Luckily, in NMR, then we could use isotope labeling approaches to overcome that issue. And so looking at the first model system, the enzyme superoxide dismutase, which has a maturation pathway from a monomer to this copper zinc bound disulfide linked dimer. So mutations in superoxide dismutase account for about 20% of familial cases of the neurodegenerative disorder ALS, and those mutations often leave the protein in this immature monomeric demetylated state that is prone to aggregation and mislocalizes to stress granules. And so for people have found that mutations of A4V in SOD1 lead the protein to co-localize with FUS, which is found in stress granules, whereas the wild type protein, which is dispersed here, does not co-localize with FUS. And this mutant A4V gives rise to aggregates in ALS. So in our model system, we're going to use caprin-1 as our scaffold protein because this is one of the major scaffolds in stress granules, but we're going to look at the last 100 residues, which are low complexity region, intrinsically disordered region that is sufficient for phase separation. And so we're going to use NMR, and we take these micron-sized condensates of SOD1 and caprin-1, and they merge together to form a giant condensate that we can stick at the bottom of our NMR tube. And this actually condensed phase has about 75% water, and the rest by mass is actually caprin-1 as the solvent. So we're going to start with N15 proton correlation NMR experiments. And if you take off this dilute phase and stick it into another NMR tube, you can measure the dilute phase NMR spectrum and look at it in blue, and you can see the amide proton chemical shift dispersion is reflective of folded protein, and the amide chemical shift dispersion for SOD1 in the Capron condensed phase is very narrow, reflective of it being disordered. So there's two possibilities: that the Capron condensate unfolds SOD1, or that the folded signals aren't detected in the viscous condensate, because we can use NMR to measure the diffusion coefficient, and we see that the condensed phase is over 20 times more viscous, similar to 70% glycerol, which is very bad for NMR because we need sharp, narrow signals that rely on fast tumbling, which is really impeded by the viscosity and giving rise to weak signals. IDRs have local chain motions that can overcome that. But for folded proteins, Louis Kay and his lab needed to come up with a whole bunch of technological advancements to enable NMR to be done on folded proteins and condensates, including using methyltrosy, which has more than 10 times the sensitivity of the amide experiments, using delayed decoupling approaches, and reconstructing signals to remove distortions, suppressing the intense signals from the scaffold, in this case Capron-1, and correcting the peak volumes due to relaxation from delays in the experiment. So getting a very quantitative measure then of the ratio of folded and unfolded states using these methyl resonances, we could see that SOD1 in the dilute phase or in buffer is less than 7% unfolded, but in the SOD1 in the Capron-1 condensed phase is over 75% unfolded. So what are the interactions that give rise to this destabilization? And we can use paramagnetic relaxation enhancement with a spin label on an NMR inactive Capron molecule and put the NMR active isotopes in the SOD1 and look to see where which residues in SOD1 are close to the spin label on the Capron-1, which we read out by broadening of the resonance in the paramagnetic case compared to the controlled diamagnetic case. And if we look at the intensities of, in this case, doing an amide-based experiment, not in the condensed phase, we can see that Capron interacts much more with unfolded resonances of SOD1 than the folded state, where we have very little broadening. Interestingly, 2 months after we did these experiments, we still had the NMR tube, and we saw that the SOD1 was aggregated in the condensed phase. And looking in with the electron microscopy, we could see these fibers, which were not present in the dilute phase and also not present in a single Capron condensed phase. You could say that the concentration of SOD1 could drive the aggregation, but in fact, Capron-1 is concentrated in the condensed phase, but SOD1 has almost identical concentrations. It's not enriched in the Capron phase, about 450 micromolar, but because of the shift in the conformational equilibrium, we actually have twice as much unfolded state in the Capron condensed phase as in the dilute phase. What about wild type SOD1? We mimicked it with a zinc loaded dimer and added a Capron to it, and you can see that this more stable dimeric metylated state is much more resistant to increases in Capron-1 concentration than the monomeric demetylated state, consistent with the wild type being not found in these stress granules leading to aggregates. But condensates aren't always a crucible for aggregation. FUS, which is another protein that is implicated in ALS, aggregates in vitro condensates. But if you remove the RNA recognition motif, which is a folded RNA binding domain, the condensates don't mature to these aggregate states. And so this RRM, which aggregates in Fuss condensates, we wondered what happened in Capron condensates. So as a quick test, we lowered the pH and increased the temperature to 45 degrees, looked at NMR spectra in the presence and absence of 4 millimolar Capron, and the RRM over time without the Capron aggregated, lowering the intensity of the resonances in the NMR, whereas with the Capron, it stayed sharp resonances, and the NMR tube remained clear for 3 months at least. So what are the interactions that drive aggregation? We did an experiment with a lower concentration of the RRM, 0.15 millimolar, and a higher concentration, 0.6 millimolar, at high temperatures, and looked at the NMR resonances. And if they interact, if residues interact with each other, we call that a hotspot, and it is expected to give rise to broadening, where we would have a lower intensity if we normalize the spectra. And for residues that are not interacting, we would expect the peaks to overlap. And what we found is that the folded resonances in the RRM largely overlapped in terms of their intensity, but the unfolded state in many cases was significantly broadened. And so we can plot this broadening, the intensity in the concentrated over the more dilute as a function of residue. And you can see these dips for the unfolded state, not for the folded state largely. And they line up with a hydrophobicity profile, suggesting that hydrophobic interactions of the RM unfolded state promote aggregation. And the next question is, how much unfolded state is there in the condensed phase? And again, we turned to the methyltrosy experiment with the delayed decoupling experiment, and we found that the unfolded state in the dilute phase was only about 8%, whereas in the condensed phase, it was over 30%. And here we actually did have differential partitioning so that the RRM partitioned more into the Capron condensed phase, so that between those two effects, we had over 8 times more unfolded state in the condensed phase than in the dilute phase. And so the question is, what's going on with the interactions of Capron? And to look at that, we did a nuclear Overhauser effect, proton-proton NOEs, looking at carbon-bound protons on Capron to nitrogen-bound protons on the RRM. And basically, all of the carbon-bound proton signals were read out, the NOEs were read out in this 2-dimensional plot to the amide protons. And if you plot the intensity of all those interactions, Capron RRM interactions to the unfolded state here, what you see is that it overlaps, not perfectly, but intriguingly, with the aggregation prone, the profile of the aggregation prone regions that I showed in the previous slide. And so the suggestion is, in the absence of a Capron condensate, the unfolded state of the RRM leads to interactions that drive aggregation, but in a condensate state, that the Capron interactions can then outcompete and actually act as a solvent. So I just wanted to then conclude and say that condensate solvent environments are increasingly recognized to play a significant role in regulating biochemical processes, biochemical reactions. for the most part, people have focused on how changes in concentration and partitioning are involved in this regulation. This can also be concentration and partitioning of ions. So you can have changes in pH, you can have changes in small molecules like ATP, and those can be regulating. But what I wanted to focus on here was this idea that multivalent allosteric solvent interactions that change the energy landscape of the proteins that are included in condensates can impact folding and unfolding equilibria and more subtle conformational equilibria that are important for active and inactive states of enzymes, for instance, binding equilibria. And these have consequences not only for enzyme activity, but also for aggregation. And I didn't talk about the translational part of this webinar, but clearly understanding this fundamental aspect that many of the proteins that people are targeting are going to be localized to condensates. We need to be aware of this fundamental biophysics in order to effectively target all kinds of proteins. And hopefully NMR can be a powerful toolkit that can enable us to access information about these conformational equilibria inside condensates. And so I'll just thank again the people who are involved from my lab, Rashik especially, and Louis, and thank you for your attention.
Thank you so much, Julie, for a fantastic presentation and thought-provoking proposal of new concepts at the intersection of allostery and condensate. We already have some questions coming in. The first one is whether you think that IDRs that do not conditionally fold can mediate protein-solvent interactions, especially in condensate formation.
Yeah, I mean, that's basically what I said in my talk. That's one of the key roles, I think, of IDRs that don't conditionally fold, is that they drive, they modulate biomolecular condensate formation to create and create solvents that then mediate conformational equilibria. Yes, yes, yes.
So, and another question is, since you showed how condensates essentially remodel quite strikingly the free energy landscape accessible to the solutes, whether in a condensate you can make the cryptic pockets that Greg was talking about even more accessible than the dilute phase. Do you think that's a possibility?
Yes. I mean, I think that the study of, the biophysical study of proteins in dilute buffer solutions has been incredibly powerful to give us some ideas about what the structures of proteins are. But inside our bodies, inside the cells, much of the time they are not in dilute solutions. So we need to think about what the relevant energy landscape is. And the relevant energy landscape is likely to be much less locked into a single narrow ground state at the bottom of those energy wells, because the environment is countering the hydrophobic effect, right? Because you've got so many hydrophobic atoms now in your solvent, so that the proteins are not going to be locked in those positions. They're going to be sampling excited states, exposing cryptic pockets, and all kinds of things, I think, with much greater frequency.
That's excellent. Thank you for your exhaustive answer. There are additional questions coming in. I think in the interest of time, we'll move on to the next speaker, who is George Lisi. but then we'll circle back to the questions towards the end of the discussion. So it's my pleasure to introduce George Lisi. He received his Bachelor of Science in Chemistry from Fairfield University and his PhD from Dartmouth College. After his postdoc at Yale with Pat Loria, he joined Brown University, where he is now an associate professor and director of graduate studies. Dr. Lisi is the recipient of many awards and distinctions, but just to cite a few, I would like to mention the Early Career Research Achievement Award from Brown University, as well as the Dean's Award for Excellence in Graduate and Postdoctoral Training. The Lisi Lab uses NMR and computational methods to study structure dynamics function relationships that are central to the allosteric regulation of several important enzymes. Without further ado, I welcome Dr. Lisi to present his talk on atomic details at the intersection of allostery specificity and inhibition of CRISPR-Cas9. Take it away, George.
All right. Thanks very much, Giuseppe. And thank you to the Protein Society for putting this great webinar on. I am excited to talk to you all today about some of the things we've been doing in our lab. Our work trends toward the latent networks aspect of the webinar rather than the cryptic pockets, but I think hopefully we can provide a nice story at the end of things. When we think about allosteric regulation in our lab, we're very interested in identifying the amino acids that might be sitting dormant within a structure that are subsequently activated by the presence of a substrate or effector to turn on networks of communication that transmit a chemical signal from one part of a protein to another. We think about this in a variety of different protein systems. In some of those systems, the pathway by which information travels is very well defined along a discrete number of amino acids in a super clean manner that you can also do MD simulations to define correlated motion between residues or uncorrelated, and you can see this in an NMR tube, in a computer, and even in a crystal structure. And in doing so, you can extract very nice in vitro and in vivo functional effects of residues along a pathway that can be involved in allosteric regulation toward a functional site, or in this case, multiple allosteric sites in a system. I won't talk about this system today. It was just a way to illustrate that in many cases, there are very, very, very well-defined pathways or allosteric pathways that exist. In larger enzymes and multi-domain enzymes, those may be more diffuse. The technique that we use primarily to decipher how these networks are connected is NMR spectroscopy. Of course, Giulia just gave us a beautiful suite of NMR data associated with a complicated protein system or a bunch of proteins. But just to reiterate, often what we're doing is taking proton signals that would come through a traditional NMR spectrum that almost everybody's seen in something like organic chemistry. We take those protons and correlate them to something in the protein, for example, the amide backbone, where we can get this two-dimensional spectrum called a fingerprint, where each peak would correspond to one amino acid in the protein. And what we're doing essentially is just using changes in either the positions of those peaks or the intensity of those peaks to extract information about structure and dynamics, and really establishing what's going on at the atomic level in response to an allosteric perturbation. And the beauty of NMR or many other methods that you've heard about already or will hear about later are that we can probe biological events on timescales that describe both the energy of the process and the relevance to allosteric regulation, which all of these different molecular processes do play a role in allostery, despite it being defined over here in this particular figure. Okay. The system that I will talk about today is the CRISPR-Cas family of effectors, primarily because it's a very large protein complex that we've been interested in for almost a decade. This is a system that is a large multi-domain protein that uses an endogenous RNA molecule to bind and localize itself along double-stranded DNA, subsequently unwinding that double-stranded DNA molecule to prime it for cleavage. What is interesting about the Cas effector is that the recognition of the DNA occurs near the C-terminal portion of the protein, but the cleavage of the two DNA strands occur in spatially separated nuclease domains, H and H here in green and RUV-C in blue. And these occur at least very closely to concerted fashion. And so the idea is that the recognition of the seed site or the PAM and the cleavage of these strands occurs in a very regulated manner with large molecular distances separating these three events. Okay. And so somehow this large protein of over 1,400 or over 1,300 amino acids, excuse me, coordinates these events in a way that is coupled to major conformational changes within this protein. Okay. One of the ways that we have chosen to study the very fine details of this, at least experimentally, has been to use NMR spectroscopy and study distinct portions or domains of this system in order to generate effectively puzzle pieces that we can piece together along with other experimental methods looking at full-length protein, functional assays, computational work, et cetera, to generate a molecular picture. We can solve crystal structures of these domains, we can get NMR structures of these domains, and we can relate them back to the structures in the full-length protein and show that they're very, very similar. I should mention here, because I have this figure up, that much of this work is done in very close collaboration with Giulia Palermo, who by pure coincidence is the upcoming speaker after me in this seminar. We've been working together on systems like this for a number of years. And so what you'll note is that in many cases, these large challenging systems such as CAS systems represent a complex that you can dissect with NMR, with other biophysical experiments, and both single domains and full-length complexes. You can couple that to computational metrics to develop allosteric models that give us residues of interest that may establish pathways. And then we can perturb those residues via mutations or other small molecules in the case of other systems, and we can determine how that effect plays out through some functional impact or some biophysical measurable. And we can go round and round in this process to take a very fuzzy picture at the large level and distill it down to a very fine detailed map. Okay, this works surprisingly well for a system like Cas9, which is very large, but we can use NMR and measure chemical shifts of alpha carbons, beta carbons, whatever you like, very accurately. And we can show on a per residue basis that those chemical shifts correlate very, very well to ensemble MD simulations of both isolated domains and the domain within full-length Cas9, right? So we can see at the residue level that what we're looking at in the NMR tube is quite similar to what we're thinking about in the large scale. Okay, and so we want to take that fundamental basis and start to think about how we can apply it to understand fundamental aspects of the allosteric process that start from an apoprotein, right, which is composed of multiple domains, but really two fundamental lobes, right? A nuclease lobe that does the catalytic reaction and a recognition lobe that binds and recognizes the nucleic acids themselves, right? And so these things work in concert. They start in this sort of open structure. They recognize a guide RNA, close down the complex. That RNA then recognizes and complements a DNA. And so as the DNA gymnastics take place, the bending and then subsequently the cleavage, we want to understand how all these different parts are talking to each other. Okay, and so this is our goal, and we'll talk briefly about each one of these things. So if we just consider this first part, right, I won't spend too much time going into this detail, but we've spent a bunch of years the better part of a decade now, thinking about the motions of this protein that go into the process of recognizing the guide RNA and communicating between the two lobes during that process. And so here I'm showing you a summary figure where basically the recognition lobe, this gray piece here that has the RNA alongside it, has all these different NMR detectable motions on various timescales corresponding to various energies, right, in various excited states that we can localize to be important in some way, shape, or form to the allosteric process. And there's many papers to describe this. And consequently, the nuclease lobe or parts of the nuclease lobe have similar motions. And I'm excluding a couple of domains here for clarity. But what we can see is that bracketing those nucleic acids, there's a lot of motion, a lot of subtle conformational changes, a lot of ensemble changes that can be detected here that are critical to this process. And they span the interfaces between domains, right? They span the apoprotein especially, this is how, in many ways, these domains talk to one another. Okay, and so one of the questions we've really been thinking about expanding that picture is what happens to that picture when you introduce a mutation that enhances the specificity of this system. So one of the reasons that Cas9 or Cas systems in general are not being used to edit the human genome, among ethical questions and otherwise, is because they can make mistakes, right? But there's a great body of literature that shows certain point mutations can be introduced into many different domains of Cas9 that essentially eliminate all off-target cleavage events, right? So that is to say, anything that is fairly similar to the target site in the genome may be cleaved by a wild type Cas, but when a mutation is introduced, for some reason, those sites are no longer cleaved, no longer tolerated, right? And so this is very interesting because in almost all cases, these are allosteric mutations, right? So these are really lacking a lot of molecular detail as to how they work. So we went ahead and thought about this. We've been thinking about this for a while. We introduced some of these high specificity mutations into Cas9, and we can look at NMR fingerprint spectra. And the one thing that I hope you can notice is that the different spectra here, colored differently, do not overlay on top of each other perfectly. You can already see by eye that many chemical shift perturbations or changes in the local environment are evident throughout the entire spectrum, right? This is a single amino acid change in a whole domain of the protein, yet we see significant changes to the structure. When we think about what these mutations might be doing, it turns out that in prior work, we've mapped allosteric pathways of Cas, and that's shown in this sort of pink color here across the H&H nuclease. So this is the domain that cleaves the target strand of the DNA, and these pink residues communicate that cleavage signal from the recognition domain, which sits over here, and the adjacent nuclease, which sits over here. And if you look at the location of some of these high specificity mutations, one of them lies directly in the allosteric pathway that we defined. And as we expect, that mutation triggers all the other residues in that allosteric pathway to feel its effect, right? Which is exactly what you would expect in a network that is all connected. But mutations that enhance specificity but are not in our pathway do not induce perturbations in that pathway. So again, this is consistent with the idea that a very specific network is being activated, but somehow these mutations are doing something too. And we were curious about that. Okay, so we started to think about this not from a structural perspective, but from a dynamic perspective. So what you're seeing here is the structure of that nuclease domain again, and all of these pink spheres here are motions that we can measure by NMR that define the pathway from the nucleic acid recognition lobe to the nucleases and the adjacent nuclease, which cleaves the non-target strand. So target strand here, non-target strand here, okay? And so this pathway of communication is NMR detectable, okay? And if we introduce, right, remember we're introducing mutations here. One of the things we found that was quite interesting is that if you do MD simulations on these proteins, you can see and capture very nicely several distinct conformations of wild-type Cas9 on its way to activation. High specificity variants suppress these off-target conformations, okay? So we have the major conformation, and then we have activated when everything is bound, right? So it's no longer equilibrating between all these sort of unproductive, if you will, conformations. This is just one metric for that. But what we were able to show, maybe more strikingly through the whole domain, is that if you think about it computationally from a network analysis, and this is work that was actually done in Giulia's lab, you can see that all of the different regions of the nuclease structure that are undergoing some sort of correlated motion with one another with respect to the whole domain, the connectivity and the way they interact with each other by the thickness of the bars and the strength of the community itself is rewired when you introduce the mutations. And no matter what, all of these high specificity mutations have a structure of communities that looks almost identical, and it looks distinct from the wild type. Okay, and so what these things are doing is they're actually rewiring how the domain is talking within itself, right? Which then propagates out to the rest of the structure in data that I won't show you here. And what's really interesting about that is that the motional change that drives this is on very slow timescales measured by NMR. So this is like a micro to millisecond timescale. Milliseconds are pretty biologically slow in some cases. And we can see that in this case, wild type protein is very flexible. The mutant as a residual has almost no flexibility in many of these key regions. But heteronuclear NOEs, which report in NMR context on faster motions, are essentially unperturbed. So they don't change. Okay, so one point mutation in the nuclease domain is affecting motions that propagate allostery outward. And if we extend that to something like the next step of the process, where DNA bending or DNA dynamics play a role, these high specificity variants tend to have more flexible nucleic acids measured computationally than the wild type. Okay, so as the protein becomes less flexible in certain regions, the nucleic acids become more flexible, right? And this makes sense because all of these things are in proximity to one another in this complex at the nexus of where the nucleic acids and the protein are interacting. Okay, and one of the things that we're doing to explore this experimentally, which we haven't published, is to look at the recognition domain in the presence of high specificity mutations and RNA DNA hybrids in the NMR tube. Okay, and so what we can see, this is just a snapshot of some of the data, is that if we look at the wild type Cas protein, or the domain that is binding the RNA DNA hybrid, let's say, and then we titrate in that hybrid, right, a short hybrid, we can see shifts associated with an interaction at many residues. But when we do the same experiment with a hybrid and a high specificity mutant, the shift trajectory is distinct from that of the wild type, right? So what's happening is that the interaction with the hybrid at the residue level is distinct from wild type in these high specificity cases. We're still doing a lot more work to kind of tease this out, but this is just a snapshot of some ongoing things that we're doing to investigate this step of the process and how allosteric mutations play a role in that. Okay, so what about this final step? DNA cleavage itself. Well, we introduced many mutations to the active site of the protein, right? And mutations in the active site, if connected to an allosteric network, should then propagate those effects. Any perturbation should be propagated out to an allosteric network that is coupled to it, right? And so when we do single point mutations of Cas residues and look at NMR spectra, again, when we overlay all of these variants, we see that they do not recapitulate the same structural environment. As expected, there are perturbations all over the place. One of the cool things we noticed, if you look at most of these perturbations, is that they're pretty linear, right? So each one of these structures occupies something in a linear trajectory, which sort of speaks to the idea that there's a conformational continuum, which is actually quite consistent with NMR studies of allosteric proteins, especially if you do this mutationally. So again, we can extract a lot of information from a simple NMR readout here. And if we think about the active site and all the perturbations that were introduced, right, here are some of the residues that are feeling those perturbations that are not part of the active site, right? So what's happening is that a change here is being felt here, okay? So again, active site sort of activating other sites, all right? And we thought about this further, okay, computationally. So in the presence of some of these mutations, which are shown here in the purple residue, again, this is some of Giulia's work in this paper that we published a few years back, what we see is that the introduction of a point mutation at the active site changes the dynamics of what's going on here and affects the proper geometry of the coordination site and therefore the DNA cleavage, right? This histidine, for example, which is catalytic, is now sampling 3 different structural states. This loop is increasingly flexible. Well, if we extend that to look at this histidine side chain via NMR, right? One of the things we did were some pH titrations to try to extract the histidine pKa here. And in the wild type case, that works beautifully, right? You can see very nice trajectories, pH sensitive trajectories as the protonation state changes, and we can back out a pKa. If we look at this mutation, we actually see 1, 2, and then 3 different substates of that histidine consistent with this molecular picture. So you can clearly see non-native states being sampled by the histidine. That's catalytic. And so what we observe generally is that we introduce mutations to the site, okay, and we downshift the histidine pKa, its basicity becomes weaker. Those with the most downshifted pKa are not active, okay? And so we're interested in understanding how this chemistry can be leveraged allosterically, right? This is orthosteric, right? But we have an idea, potentially, that if we can modulate the histidine pKa we can chemically supercharge Cas9, right? One of the things about high specificity Cas systems is that their activity tends to be lower than that of the wild type. So if we were to come up with a recipe for something that we were rationally designing allosterically, we would want to increase the basicity of the histidine. We don't want to sample minor states at the active site. We want to minimize perturbations to the chemistry of other residues, and we want to keep the biophysical markers of high specificity. Okay, so when we do this experiment for wild type, we can again see very clean trajectories. We can fit pKa values very simply. But if we go back to those lysine mutations that I talked about a couple slides ago that induce high specificity Cas, we immediately see that we have these off, these minor state populations of the histidine forming in addition to our major trajectory. So we get multiple states of this histidine catalytic, though we do upshift that pKa. If we make that alanine now a glutamate, we can sort of minimize the existence of the minor state. We still upshift the histidine pKa, but let's just say we're working on this, right? So we're introducing lots of different allosteric perturbations in the pathway that we've identified all those years ago to see if we can do something to modulate the chemistry of the histidine from an allosteric perspective, okay? And to show that this is something that's relevant to the allosteric pathway of the nuclease itself, we introduced a mutation that is not in the pathway and nothing happens to the histidine, right? It doesn't change any of the pKa's or anything like that. And one of the cool things is that, of course, Julie just spoke, and Lewis have an experiment that they published many years ago about using pH to assess aspartate pKa's. And we can use that experiment to show that the pKa's of most of the aspartic acids don't change despite these mutations. So we're changing the histidine, but not the adjacent aspartic acid in that site. Okay. So in the last couple minutes, I just want to mention one quick thing, is that in addition to mutations in allostery, we also are interested in exogenous ways to do this. One of the ways to think about this is through a phage-derived anti-CRISPR protein. So these have evolved along with CRISPR systems to prevent bacteria from killing phage that invade them, right? So you could imagine that an anti-CRISPR protein could bind to a Cas system and inhibit it, and therefore the phage could invade the bacterial cell and survive rather than being destroyed. So it turns out there are a couple of these anti-CRISPRs that target many Cas domains. They have orthosteric and allosteric interactions. We solved crystal structure of one of those. It's an orthosteric interaction at the catalytic histidine. But the NMR perturbations associated with that occur not only at the binding interface, but they propagate out into the allosteric network. And we can very clearly see the population of the free state decrease and the bound state go up, and nice slow exchange tight binding NMR. And if we introduce a mutation at this interface, we lose the ability of the bound state to fully populate. Okay. There's an orthosteric effect, as I just showed you, between the direct interaction where the histidine and associated residues near the active site are pulled out of plane when we look at two crystal structures. In green, the H and H alone, and the pink is the ACR bound. But there's also an allosteric effect at the core of the protein where you get a lot of these different dynamic aspects of side chain residues that are changing position in response to the ACR, even though they're not interacting with it directly. And this is really cool because what the anti-CRISPR does is it stimulates NMR-detected millisecond motions that are not present in the apoprotein. And while that's happening, fast timescale motions in the same areas of the protein are suppressed, right? So we have a decrease in the fast timescale bond vector low energy fluctuations and an increase in these higher energy slow timescale motions in sites where they did not previously exist. So what the ACR is doing is structurally and dynamically modulating this. And of course, it prevents cleavage. We see a lot of uncleaved bands, although a little bit of cleavage in preliminary stuff. But if you introduce any of these mutations at the binding interface, you essentially have no starting material left. You have almost all cleaved stuff, so it can't inhibit anymore. One of the cool things is that this anti-CRISPR, though it binds to the nuclease, also lowers the affinity of the whole Cas protein for its guide RNA, which does not interact with the nuclease in any way. And so it is propagating some sort of effect that we have not yet teased out to that to that domain. And what's really cool is that the anti-CRISPR, if you think about it in the context of a full structure versus our isolated structure, it actually clashes with the nuclease, suggesting that it actually wants to bind to an allosteric region of H and H, not orthosteric. And if you form the Cas9 RNA complex before you add the inhibitor, it doesn't inhibit, right? So it has to block a certain conformational change, but once the RNA is present, the conformational change has already happened, and the anti-CRISPR can't occupy that orthosteric site. So that means that a model in which allosteric binding of these anti-CRISPRs at a weak affinity might be favorable if any type of nucleic acid is already bound, right? In the apo state, it can occupy this orthosteric site, but when bound to a nucleic acid, it wouldn't inhibit effectively unless it was bound somewhere else. The last thing I'll say is we asked the question if there's any evidence of this, and the answer is yes. If you titrate an NMR spectrum, if you titrate anti-CRISPR to significant molar excess above the initial binding event, you see very specific NMR changes, chemical shift perturbations. This is after all of the stuff has already happened in response to the primary binding site. And so these shifts map to different regions of the H and H domain that are not the primary site, right? And AlphaFold actually predicts two anti-CRISPRs bound to this H and H site, one of which lines up with some of those NMR chemical shifts. We can fit these shift trajectories to show that the binding is actually pretty weak and therefore fairly transient. And we can see that the histidine side chains, including this one that's over here, are shifted and line broadened in response to the anti-CRISPR, suggesting there may be an interaction here. So we're trying to get more experimental information about whether this complex actually exists. The last couple slides, I just want to mention some things that we're doing to capture larger portions of Cas9 allostery. As Julie mentioned, methyltrosy is a great metric to assess structure in large biomolecules, so we can take larger chunks of Cas9, like the whole recognition lobe, and do methyl NMR and get relaxation experiments out of this. There's also a really nice paper that was published a couple of years ago almost about using methyl experiments to get almost whole Cas9 stuff. And at NMR-FAM in Wisconsin, we're doing some solid state on the whole protein. Here's some really beautiful spectra. This is whole Cas9 in a solid state rotor. And so you can get really nice spectra of that. So we think we'll be able to get a lot more information about the long range effects in all the domains from solid state. And the last thing that we've done is some HDX mass spec, which tells us about deuteration patterns. And so this is in effect a dynamic, peptide level dynamic experiment where Cas9 in the presence of deuterium over time, can show that its guide RNA protects it, right? There's almost no deuteration here. It all lies in the apoprotein. And so guide RNA compacts the structure and suppresses this HD exchange across the whole protein. But if we introduce a high specificity mutant with mutations in this domain, we can clearly already pick out changes that are distinct from wild type across the whole protein system. And so as we look at deuteration patterns where, we see shifts for deuteration and protection from guide RNA, we can do this in high specificity mutations too, and localize changes that occur. Mutations are here, the effect is here, right? So this is something that we're really interested in furthering down the road. And we're extending this to different systems like Cas12a, where we can introduce mutations in unstructured regions between nucleases that don't have any functional consequence, but they don't change bulk secondary structure, they don't change stability, they don't change RNA-DNA hybrid affinity, but they don't cleave. So we're really interested in teasing these things out as we go. And so I'll stop there and just say thanks to my lab and the people who did the work. And thank you very much again for the invitation.
Thank you, George, for a great presentation. Very nice delivery. We already have a question. Have you compared the residue networks identified in Cas9 using biophysical models to covariation or evolutionary couplings?
Yeah, excellent question. To some degree, I think that we need to do a more rigorous assessment of the experimental networks in the context of covariance and the evolution in order to extract out what we want to say about the pathway extending beyond a single domain, right? So I think at the single domain level, we know a lot of information about the pathway because it's easy to perturb and really test the outcomes. But I think in the context of the whole molecule and how the information gets, especially from the recognition part at the C terminus down to the nuclease, that's where I think we have some blind spots and we really need to do this type of stuff. So that's a really good suggestion.
And do you find that your allosteric pathways tend to be bidirectional?
Excellent question. In the nuclease that I talked about, yes, right? Because if we perturb almost any site along there, we see the functional effect. And if we perturb the active site, for the most part, we see that reciprocated. In the recognition domain, which binds the nucleic acids, we see only some reciprocity, and it tends to be in the residues that are close-ish to the RNA itself or the RNA-DNA hybrid.
Okay. Thank you so much, George. In the interest of time, we move on to the last but not least talk of the webinar by Giulia Palermo. Giulia received her bachelor summa cum laude in chemistry and pharmaceutical technologies from the University of Salerno and her PhD from the Italian Institute of Technology. After a postdoc at l'École Polytechnique Fédérale de Lausanne in computational biochemistry with Ursula Rotlisberger and a postdoc at UC San Diego with Andy McCammon. She joined the University of California, Riverside, where she's now a professor of bioengineering. Giulia is a Camille Dreyfus Teacher Scholar and has received several prestigious honors, including the Corwin Hanash Award for Outstanding Scientist under 40 and the NSF Career Award and the Sloan Research Fellowship in Chemistry. Dr. Palermo is best known for her pioneering studies of the CRISPR-Cas9 system, for which she has developed and applied cutting-edge computer simulations and artificial intelligence AI approaches. Without further ado, I welcome Dr. Palermo to share her screen and present her talk on advancing gene editing through computational methods and deep learning. Take it away, Giulia.
Thank you very much. Good morning, everyone, and I would like to thank you very much for the kind invitation and for this opportunity to report here some of the research we are pursuing in my lab. So, as mentioned, my lab is interested in studying gene editing systems through computational methods. And today I would like to report some of the new results integrating artificial intelligence approaches with molecular simulations. This is the overall goal. This is to introduce the overall goal in my lab, where we started with computer simulations of CRISPR-Cas9, where we are now at the interface with artificial intelligence, and where we hope to be in the future to develop and apply computational methods to perform personalized medicine into the long run. We started studying CRISPR-Cas9, and which was beautifully introduced by George, and the way actually how we like to say CRISPR-Cas9 work, we like to say this is a protein that works a little bit like an aerial silk artist. Is a protein that grabs the RNA, changes conformation to bind the DNA, match it to the RNA, and perform cleavages. And of course, along this beautiful dance of the protein, we have a series of open questions, such as conformational activation, catalysis, off-target effects. All of them we studied through computation. But today, I would like to focus on the allosteric signaling of Cas9. So we started this simulation, this project, employing methods that were previously applied to study allostery in protein-drug interactions. So a more canonical way to see allostery. In particular, graph theory approaches in which you can describe each amino acid as a node connected with edges, and these edges can be weighted by a property, some in our case, correlations. The way how you can see these methods is a little bit like when you are on social media, you have different friends, all they are connected. Some friends hold more connections than other friends, which clearly makes you not wanting to tell your secrets to these friends, because these are the friends through which the information passes in a more efficient way. So these are the nodes of the network that we want to classify within our simulation system. And through these approaches, we can also calculate the shortest pathways of information transfer from one site, one desired site, to the other. For example, this is our Cas9 protein. We have two catalytic domains, so we can calculate the shortest pathways of information transfer, the one that maximize correlations from one site to the other through these algorithms, for example, the Dijkstra algorithm. And this is what we have been doing in collaboration with George. We had several papers in collaborations with George. In particular, we wanted to characterize what are the shortest routes of information transfer between the sites of DNA recognition and the sites of DNA cleavage. The sites of DNA cleavage. So our approach, our shortest pathways overlapped with residues that were observed via CPMG relaxation dispersion to be slow, particularly prone to transfer the allosteric signaling. By further computing the shortest pathways of information transfer between the two catalytic sites, we identified residues that we proposed are central for governing the dynamics of the system. Based on these findings, there have been modifications of the enzyme attempting to improve the specificity, and as you can see, it has been shown by these groups that by altering these transducers, these allosteric transducers, the specificity of the enzyme can be improved. So this has shown that there is a clear connection between allostery and specificity in Cas9. Always in collaboration with the Lisi lab, we went further in understanding how mutations located in the recognition lobe, so farther away from the HNH nuclease lobe, alter the dynamics of the HNH site. And we reported in this paper that these mutations distally located with respect to the site of cleavage increase the communication between a very important helix in the recognition lobe, which is this REC3 helix, which was previously shown to recognize mismatches within Cas9. Another chapter of our studies involved the study of the catalytic mechanism of DNA cleavage. Cas9 holds two active sites, HNH and RAVC, which concertantly cleave the two DNA strands. And to study how Cas9 cleaves DNA, we used mixed quantum classical approaches. So the active site is treated at a quantum mechanical level, the rest of the system is treated at a classical MD level. Of brevity, today I will focus on the study of the HNH catalysis. We have studied how HNH cleaves DNA by the use of QMMM simulations and an extended Lagrangian metadynamics approach, which allows us to recover the free energy surface using QMMM simulations. We have revealed that the catalysis is activated by this histidine, Trogoutly studied by George through NMR studies, and is basically an associative mechanism in which this lysine is actually very important for the completion of the catalysis. But the best way to watch the catalytic mechanism is through a movie. Here you can see our system. This is CRISPR-Cas9, very large system. To my knowledge, this is the largest system that was studied with QM ab initio molecular dynamics. The spliceosome was also studied, but a smaller model was used. So this is, to my knowledge, the largest that was studied so far. And here you can see how the histidine deprotonates the water molecule. We reach the transition state where bonds are partially formed and broken. Then the schissyl, the bond breaks, and the catalysis is completed by a proton shuttle through this histidine. A postdoctoral scholar in my lab, Mohamed Ahsan, has built on this study to characterize how alternative metal ions, for example, magnesium, aluminium, lithium, cobalt, affect the catalytic mechanism and the activation of Cas9. In particular, Cas9 changes conformation from an inactive state in which HNH is far from the cleavage site to the active conformation in which the catalytic histidine is prone to perform cleavages. Single molecule experiments have shown that by changing the concentration of metal ions, in particular magnesium, calcium, and cobalt, by changing this concentration, you can reach easily the active site. So basically, the HNH domain assumes more favorably an activated conformation. So we decided to study the thermodynamics and the kinetics of this conformational change in the presence of high and low concentrations of magnesium. And clearly, in the presence of high concentration of magnesium, this conformational change is thermodynamically favored, but we also observe through Markov state modeling that it is 2.5 times faster. Which shows that magnesium has also a structural role, not only a catalytic role. But what was more interesting of this study by Ahsan, Alexa, and colleagues, is that they were able to observe the formation of a cryptic divalent metal binding pocket at high concentration of magnesium. A pocket that is not observed in cryo-EM studies, is not observed in X-ray crystallography studies, but transiently appears at high concentration of magnesium to enable HNH to more favorably reach the active site. Instead, in fact, sorry for my English, at high concentration of magnesium, HNH reaches more favorably the catalytic pocket. And this happens because magnesium locate into this pocket that transiently forms at high magnesium concentrations. This is something that was further confirmed by the work of George Lisi and David Taylor. In particular, as we know, it has been beautifully introduced in the first presentation today by Greg Bowman, that these pockets are called cryptic. This name cryptic is a name that is given because of the fact that these pockets are transient. They cannot be observed in the time scale required for capturing the system through X-ray crystallography or cryo-EM, but since they are cryptic, so transient, their timescale allows studies through solution NMR that can capture very fast and slower timescales. Here, through CPNG, relaxation dispersion, and through chemical shift perturbation, we were able to observe differences at the level of the cryptic pocket. And also important, while mutating these residues, the catalysis of the target strand is hampered, showing that, counter-proving that the formation of this cryptic pocket is very important for the activation of HNH for cleavage. The second part of my presentation will focus on a new project that we are pursuing in my lab, a different project that involves with gene knock in rather than knock out. So what Cas9 does is to introduce knocks out, so cuts pieces of DNA away. But what we are focusing now is how to knock in very large pieces of DNA within the genome through DNA transposition. DNA transposition was already known in biology from the 80s, when the Maids Queen was able to find transposons in maids. But very recently, a new paradigm in life sciences appeared. The idea that DNA transposition can be guided. So big pieces of DNA that, according to what was known before, can jump in the genome from one location to another, now they can be targeted at specific positions of the genome through specific bacterial systems, always coming from the CRISPR world. Of course, this is absolutely exciting for us, but also represents an incredible challenge for molecular simulations. The reason why there is this incredible challenge is because these systems are composed of several proteins. They are extremely large, so very challenging for computational resources, but they are characterized not only by conformational changes, but also compositional rearrangements, which makes the computational approach particularly difficult, but also particularly exciting. Here is, if we look at the structure of the transpososome as obtained by our collaborator Elizabeth Kellogg, we can see that the transpososome is composed of a recognition complex made of a Cas protein, so Cas9, S15, and TNIC. Then we have these proteins called TNSC that polymerize until they find the binding of TNSB, a transposase enzyme that brings the DNA that we want to insert within the DNA and inserts through cut and paste the DNA at a specific position, establishing the cleavage, the insertion site. So here you can also see it much better, the process. Here you have the CRISPR-Cas complex, you have additional transposition proteins. This is the transposase that brings the piece of DNA that we want to insert at a specific site. And the first question we wanted to address, the first system we decided to study is the type 1F3, so-called cascade TNIC complex. Cascade, this complex of multiple Cas proteins in complex with TNIC. Here, into these cryo-EM structures, the author were able to obtain this very beautiful structure, but because of the mobility of this region, the high mobility of this region, they were unable to fully characterize this portion of Cas8, which we called Cas8 bundle, was called in the literature Cas8 bundle. So what we did was to use AlphaFold to reconstruct this alpha helical bundle. And what was very interesting is that AlphaFold suggested two possible conformations, an open conformation and a closed conformation. While the closed conformation was very similar to the cryo-EM density, fitted in the cryo-EM density, the open conformation did not. But AlphaFold provided equal probability, equal possibility for the two conformations. And according to what was reported in the biochemical literature from other CRISPR-associated transposons, an open and closed conformation could be possible. So we decided to study this conformational change through free energy simulations, and we observed that as the DNA fully binds the guide RNA within the system, this conformational change is much more favored, suggesting that the purpose of this conformational change is to hide the binding of DNA to the system. My student Aman Patel also decided to use an approach coming from physics, in particular wavelet analysis. So basically what he did, he took the displacements from the simulations, he converted them into wavelets, and he was able to calculate these plots in which we can characterize what is the scale of the motions in terms of little waves. And what you can see from these results is that when the conformational change happens in the presence of the longer DNA strand, we have coupled motions of this conformational change with the other motions of the systems. So the conformational change is synchronous with the movement of Cas9, but in the other case, instead, we have cacophonous motions that are not synchronous, sorry, with the conformational change of the bundle. Finally, I would like to introduce another question that we are in my lab about DNA transposition. This is the type 5K system. In this system, we have our recognition complex, TNSC, polymerizing around double-stranded DNA until it finds the transposase for cleavage, for DNA insertion. Here there are key questions we want to address. How does TNSC bind to DNA? How does TNSC elongate along double-stranded DNA? Does it elongate in the 5 to 3 on the 3.3 to 5 direction? This is a key open question in the field. And what is the effect of TNSC binding on DNA? In particular, this question is very important to address this question because there are in the literature several TNSC, some of them distort actively DNA, some of them do not. So this is also a conundrum in the field. So we performed molecular simulations of a very long filament of TNSC bound at TNA, and we observed that in the presence of TNSC, we have a distortion of the DNA, as you can observe in different DNA parameters, minor, major groove, DNA length. The DNA is remarkably longer in the presence of TNSC. We also observed that TNSC establishes very critical interactions with the backbone between these particular residues that, if mutated in alanine, hamper DNA transposition. We also looked at the dynamics, more broadly, what is the dynamics? Sorry, just a moment. What is the dynamics of TNSC in the filament? Here you can see from cryo-electron microscopy an analysis of the dynamics through the principal component. This was made by Elizabeth Kellogg, and here you can observe that the largest motions affect the peripheral area of TNSC, in particular the C and N terminal, which reflects what we observe through principal component analysis. This is the first principal component from simulations. But to fully address the question whether TNSC causes the dynamics of DNA, so causes the distortion in DNA, we decided to use causation inference models, and in particular Granger causation. Granger causation is an approach developed in econometrics and is a statistical test used to determine if the past values of a time series can predict future values of another time series. The method is also applied in systems biology, and in this case, we use Granger causation to assess the directional coupling between dominant protein motions and DNA remodeling, to test whether the fluctuations along the first principal component, Granger cause, the DNA minor group width opening, the major group width opening, and the change in the backbone RMSD of the DNA. And as you can see from this analysis, what is particularly interesting is that the ISM linker 2, they directly Granger cause the distortion of DNA, as well as the terminal regions, which we can see from principal component analysis of the experiment and the computation, are particularly relevant. So there is, we can infer that TNSC causes DNA deformation through this approach. But now we also wanted to address how does TNSC bind and elongates on double-stranded DNA. So here, two key questions we want to address. Question number one is, how does TNSC first nucleates on DNA? So how does it first bind in the presence and absence of ATP, and what is the role of ATP? The second question we want to address is, how does the filament form? How does it elongate in one direction or the other? So to address these questions, we used free energy method. So what we did, we placed one TNSC far away from DNA, and we studied how TNSC binds on DNA in the absence of ATP. Clearly, we see that in the presence of ATP, the system reaches the minimum of the cryo-EM structure, which instead is not observed in the absence of ATP. And this is basically happening because, as you can see, TNSC is much more rigid in the presence of ATP. So ATP also has the important role of holding, conferring to TNSC the right rigidity to be able to reach DNA and the minimum of the free energy, which corresponds to the cryo-EM structure. What is very noticeable in our simulation is this nucleation point. This nucleation point, this is a point, the point of nucleation was defined because at this point, in the presence of ATP, the DNA, the DNA minor groove starts widening. And this is the reason why we referred to this point as nucleation point. So the point when TNSC starts binding to the DNA and starts inducing this widening of the minor groove. Why this is important? Because at this point, we observe several interactions at the level of the minor groove that open DNA, but the reason why they are also important is because recent experimental studies by Kellogg show that mutations of these residues are critical for the specificity. So these simulations reveal not only how TNSC nucleates on double-stranded DNA, but also reveal that the specificity is established at the nucleation point when these residues bind the minor groove rather than the backbone, which is observed at the level of the cryo-EM structure. So what happens? TNSC binds, first reaches the minor groove where the selectivity is established, selectivity for A, T versus GC, and then these interactions change, they move toward the backbone for stabilization as observed into the cryo-EM structure. Finally, we wanted to study the mechanism of elongation. How does TNSC, once bound at the level of DNA, keeps binding along double-stranded DNA in one direction or in another? So here we performed again free energy simulations using, in this case, umbrella sampling simulations, and we observe that in one direction it is slightly more favored than in the other direction. The 5-3 direction is slightly more favored, which is in agreement with the experimental experimental studies by Park and Kellogg, and this was work that was performed by Chinmay Pindi, another postdoctoral scientist in my lab. Chinmay, together with Ahsan, introduced the use of graph attention network for the analysis of these simulations. Greg has already introduced graph neural networks and the use of attention for the characterization of cryptic pockets. Here we want to use the attention mechanism to characterize the regions of the system that drive classification. So we have our CRISPR-associated transposon, we describe it as a graph of nodes and edges, and through the graph attention network, we build graph attention networks that are able to classify different conformational states, and most important, are able to derive attention scores. So attention scores that can characterize regions of the system that are critical for the classification decision, that are critical to lead to a specific conformational change, resulting into the characterization of different conformational states. The way how you should see graph attention networks, you have to imagine the protein like composed of a series of presenters in a poster session where you have different posters and several people around each poster. But of course, the presenter has the biggest attention from all the other. Graph attention network enables you to characterize what is the actor that holds the highest attention. And in the case of a protein, this actor that holds the higher attention is a region of the protein, a region of the DNA that drives the classification decision. So we applied this graph attention network to characterize, to understand why one transition state is more favored with respect to the other. So what we did was to classify the two transition states and extract the attention scores between the incoming monomer and the bound monomer, the various regions of the bound monomer and the regions of the incoming monomer in 5 to 3 and 3 to 5 directions. So clearly from this analysis, what comes out is that in the 3 to 5 direction, the unfavored, we have several attention, a number of attention with high value, which signify strong interactions, a variety of strong interactions that keep the system in the transition state longer. While in the other case, we can observe fewer attention, but one of them has very high variance as depicted through the thickness of this plot, of this arc plot. High variance in this case signify interactions that are forming and breaking transiently, enabling the transition state to be shorter, favored, and falling easily in the product. An analysis of the attention network shows us that at the transition state in the favored pathway, we can see that these two regions, linker 1 and the N-terminal, bind together. While in the 3 to 5 direction, we have different regions, multiple regions that bind together. Why this interaction is very important? The reason why this interaction is very important is because here we are not in the cryo-EM state. Here we are at the transition state, when the two systems encounter each other, first encounter each other, and then change conformation to reach the cryo-EM state. You can see how the systems encounter, these two domains encounter each other from a movie of the simulation, showing that when we reach the transition state, linker 2 and N-terminal of incoming and bound monomer encounter each other and change conformation when we reach the cryo-EM state. So I would like to dig a little bit more on why this information is so important. This information is very important because there is still a debate in the literature. There are some papers in which they show experimentally that the elongation of TNSC is favored in the 5 to 3 direction, while other papers state that the elongation is favored in the 3 to 5 direction. However, these papers differ on how the experiment has been done. When the experiment is done only on a single DNA, the 5 to 3 direction is preferred. But as other transposition components, such as Cas12, is bound, the 3 to 5 direction is favored. According to our data, it's very important to keep in consideration that the pre-bound monomer or the pre-bound subunit that could be Cas12 or another transposition component influences the formation of the transition state. In fact, in 5 to 3 direction, you can see different interactions at the transition state with respect to what is observed in the 3 to 5 direction. In the same way, if we have an additional component, which could be Cas12 or the other transposition components, we might have a different mechanism of nucleation and elongation, questions that we hope to test in our next paper. And this is the mechanism that we have proposed for the elongation in the presence of double-stranded DNA only. So TNSC binds DNA through the ISM region, and this is the first monomer that nucleates. The second monomer binds through the interaction between linker 2 and the N-terminal. The system changes conformation, reaches the cryo-EM complex. As in this state, the complex is now ready, exposing the N-terminal for another subunit of TNSC to bind through linker 2 and N-terminal, and the elongation continues. Finally, I would like to highlight this other recent work that we have contributed with Liz Kellogg. So she obtained this structure of a new type 1F3A Cas transposase. Here we contributed understanding the role of IHF, an additional protein that is very critical to bend DNA and enable binding of DNA to this system. And with this, I hope I have convinced you that we can properly combine molecular simulations and artificial intelligence, new artificial intelligence approaches to characterize gene editing systems, and in particular all these new exciting systems that manipulate DNA and nucleic acids in a way that is really unique and is transforming the way how we look at biochemistry. And with this, I would like to thank all of you for your attention, and of course my lab that so incredibly performed this research, was so incredible and so passionate about this research. And all of you for your attention. My lab is currently moving to UCLA, and we hope that we'll be able to pursue exciting science.
Thank you so much, Giulia, for a very impressive presentation. I would like to thank all the presenters of today. In a way, allostery in terms of free energies is a form of non-additivity, but we can also say that for today's webinar that the whole is greater than the sum of its parts, where the parts are not only the presenters, but also the online participants that stimulated the wonderful discussion with their questions. And in the interest of time, I would like to open the panel-wide discussion at this point. And I do have some questions, perhaps starting from the cryptic pockets, Greg. So my understanding is, provided that you go high enough in the free energy landscape, sooner or later you'll find an excited state with some open cryptic pocket. However, It's also true that the effective affinity for targeting the cryptic pocket is scaled down according to the population of that excited state. So there is a trade-off between the population of the excited states and how good the pocket that you find higher up in the free energy is.
Yep.
So what is your suggestion on how best to strike a compromise between these two opposing tendencies?
Yeah, so my very somewhat snarky but practical answer would be that if I can see it in simulations, it's populated enough to be useful, right? And just to, a more quantitative, I would say that even states that are like 10 kcal per mole above the ground state are viable, which is quite high. And this comes from looking at the apparent affinities of known drugs range from 5 to 15 kcal per mole, and that's mostly against, pockets in the native state, right? The ground state, I'm going to assume, right? So that means, 5 is potentially good enough. So something that's, 10 kcal per mole, if you have a binder that's, getting 15 kcal for that state, you could shift the whole landscape to be relevant.
Right. The trade-off would still be very favorable.
That's right.
Thank you so much, Greg. And in light of what Julie explained about condensate making, at least in some cases, excited states or some partially destructured states more accessible, are there ways to model excited states within condensates?
Yeah.
Or cryptic pockets within the condensates?
Yeah, I think so. I mean, in principle, we can model anything made out of atoms, right? It's really just a matter of how long it will take us. But
You can model a human being.
yeah, that's right. I mean, there's people simulating chunks of the cytosol, right? So I think given that, it should be possible to like take those sorts of setups and look at cryptic pocket opening in that environment, for example.
Well, can I just jump in and ask Greg?
Yeah.
So, a lot of the simulations of condensates have been in coarse grain models. And I'm just wondering if the coarse grain model paradigm is sufficient to get the information on cryptic pockets.
I have healthy skepticism, but I'm glad if people try. I think some of the new things with machine learned coarse grained force fields have the best potential because it can get implicitly, I think, more of the complexity than assuming everything is globs of atoms or spherical, for example, right? So it'll be interesting to see.
Yeah.
I also had sorry, Julie.
No.
I also had kind of a open sky hypothetical question for everybody in the panel. Like assuming that funding, NMR time, computational resources were not an issue, very hypothetical. What do you see as the main unsolved problems and challenges in your field? Perhaps Julie, do you want to go first? I know your schedule is quite tight.
I'm gonna say there, I could talk from here till next week of the unsolved challenges, but the ones that are relevant to this webinar, I think that characterizing conformational equilibria inside condensates is a driving need, because if we're going to target proteins that we think we know what its structure is, where that structure is actually perturbed inside cellular condensates, then we have an issue, right? So I think this is an unsolved problem. And, the modeling efforts are usually in toy systems like we do our NMR with 100 residues of caprin as modeling the environment of a stress granule, which is a complete, joke, right? So how are you going to model the hundreds of proteins that are in a stress granule as the actual solvent environment for all the proteins that are in there, and then think about what the conformations are, the conformational equilibria that are involved in enzymes, the binding equilibria, how they're impacted by that new solvent environment. I think that's it. That's, a huge computational and simulation challenge and experimental challenge as well. In vivo NMR, maybe in some dream world. Yes.
Thank you so much, Julie. Anybody else wants to chime in on these grand challenges?
Say the one that I'm going, well, given the pressure to be of practical utility at some point, right? One of the things I'm most obsessed with is, how do we know if we've successfully hit a cryptic pocket, right? Because if you do an activity or function assay and see a difference, with something you meant to hit a cryptic pocket, it can be hard to know if you succeeded, apart from solving a crystal structure, which is not something you're guaranteed to be able to do, right?
I think your hypothetical of unlimited experimental time. I mean, I think it's things like looking at transient disease states and their connection to allosteric networks in at least in things that we think about, or like continuously, even though people have been pitching it for quite a while, looking at the biggest systems possible, pushing the experimental capabilities of atomic biophysics as high as possible, if we don't have to worry about how much time and effort it might take to extract some of these things, because it's like the interface of complicated networks and transient nature and ability to spend time collecting the data to get it seems like still a weird axis to be at.
Thank you, George. Great points. I
Yeah.
guess, Giulia, you wanted to chime in as well?
I completely agree with George and with Greg. So I think that the interface between different techniques is becoming more and more important, right? So as we, for example, in the case of cryptic pockets, as Greg also has shown, computations have been very important to characterize these cryptic pockets and have helped experiments dramatically, but at the same time, experiments are required to test the existence of this cryptic pocket. Like in this case, NMR is really a very optimal tool because of the time scale that NMR can access and the fact that it is in solution. And as we move forward, we always want to do new things and apply new techniques and study, come out with new exotic ideas, right? So we are really excited about all these new neural network diffusion models to see how we can use them in biophysics, not only for design, but also for biophysical understanding. And personally, I'm very interested in understanding how the artificial intelligence tools work. So not only for a mere application, but also for understanding how they work and how we can tune them for our purposes, not only for design, but for mechanistic understanding. And also I'm very interested because of training the students in these new methods.
Yeah, thank you, Julia. That's actually a very interesting point because certainly AI-based modeling has very strong predictive power, but when it comes to interpretability, still we have to go back to more traditional science, trying to understand why some very interesting AI-enabled predictions are actually checking out experimentally. Do you think that these AI models are just a purely statistical exercise, or in the process, the AI models are actually learning a little bit of physical chemistry.
Yeah. So actually, this is a very interesting question, and thank you very much for doing this question, because I went through the attention model, and the softmax, just the equations of softmax is the probability distribution. So basically, these approaches, the neural network inside, if you read very carefully how the neural network is organized, the neural network itself, plus then graph attention model and all what has been done, if you read this carefully, you will see that these are all rules coming from statistical physics, statistical mechanics. So this means that there is an intrinsic interpretability that we should be able to decipher if we go inside carefully, which is very interesting in my opinion. And also the diffusion model from the Baker lab, if you read the supporting information, you'll see that it's basically matricial algebra and other statistical approaches that we can decipher through the analysis of by reading carefully how the model is built, and then we can take, for example, one approach, mix it with the other, and so on, is a field that is really interesting. I think that it came out from now is coming all this AI, right? Everybody's talking about AI, but the way how it was developed was really through the intersection of fields, which is one of the most exciting things, in my opinion. Applied mathematics and statistical thermodynamics, then molecular. Now we can interface this AI coming from applied mathematics, statistical physics with our simulations, and of course with the experiments. It is all very exciting, and understanding how it was built to me is very interesting and is very important as educator for our students to make them aware of how these new technologies work.
Thank you, Giulia. Now, I mean, the title of this webinar is Translational Allostery. So ultimately, all this wonderful science has to translate sooner or later into some clinical impact. And I mentioned at the beginning the example of allosteric inhibitors of ABL tyrosine kinases for leukemia. What do you guys see as the next deliverable in our field of allostery? In your domains, for example, maybe a new mutant of CRISPR-Cas9 that simultaneously optimizes specificity and activity.
I mean, those cases, like, things that are manipulating nucleic acids, I mean, there are commercially available Cas systems that do great things, right? They're very specific, they don't have off targets, etc. I guess the thing would be to understand the rules by which you could make your own, right? And you could customize it to anything you wanted. And, aside from error-prone PCR, or aside from, a directed evolution pipeline or something like this, where you could look at it and just say, this is what I need to manipulate because I understand how the network works, or something like this. And that could be, across genome editors, it could be across base editors, things like this, right? Those rules might all play a role in similar systems that interact with nucleic acids, or DNA repair or something like this. They could all be leveraged in a similar way, maybe. So that's one thing in terms of that aspect.
Thank you, George.
For you all, how close are we to that? How close? I think we get some things. I think we really do. I think we're way better than we used to be, but we have stuff to learn for sure. But I just wondered how close. What problems are truly tractable, right? We know that what Julie defined here is not, I mean, that there's a lot of work to do there. But some things I think we get better. And the interface between the experiment and computation is ever more, I think, helping.
I think some of the new things with protein design are super exciting. I mean, we've got some I think, pretty compelling stuff coming together, showing we can use that to intentionally hit our cryptic pockets, right? And like that's a good step towards proof of concept. And
Yeah,
But I
that was
think,
not a thing.
yeah.
So that's an amazing advance. Yeah. Sorry.
And
Yeah.
Greg, no, sorry. Greg, you also had some examples for infectious diseases, right? I think in your talk.
yeah, that's right. I mean, we've done a bunch with coronaviruses and with Ebola, right? And our Ebola test case is the one where I'm pretty confident we have peptides that hit our favorite cryptic pocket now.
And how sensitive do you think are these cryptic pockets to viral mutations?
Oh yeah, so that's one of the exciting things too, I guess, getting to the understanding side of things that George said is that we've shown that that cryptic pocket has a function, right? So I think it's actually under selective pressure and that there's a balance between the population of open and closed cryptic pocket states that's essential, right? And so that's cool because either way we can push it is useful therapeutically.
Thank you, Greg. Okay. There are still some questions left in the Q&A, mainly about condensates. So, Julia, I don't know if you have time, but one question is about how to differentiate between condensates and aggregates.
So fluidity, an aggregate is a solid, and condensates can be very, very viscous, even to the point of some people saying they're solid, but they're defined by their solvation. So their molecules are coming in and out. So that's the difference between an aggregate and a condensate.
Okay, thank you, Julie. And then there is another condensate question. Very nice talk. If condensates can be thought of as effective solvent environments, is there a condensate composition for which crowding and confinement effects outweigh specific enthalpic interactions so that proteins become more folded rather than more unfolded? Could different condensates therefore be classified according to an effective solvent quality that determines their impact on protein stability?
So I think that's very true. And so the condensates that are formed by complex coacervation of highly charged disordered regions, that is going to be a very different kind of a condensate than a condensate that is formed by IDRs that have a lot of aromatics and pi groups and hydrophobic groups. So yes, I think that there are going to be unique solvents that are going to have different impacts on the energetic landscapes. So yeah, my generic sense that condensates are going to always lead to some sort of destabilization is probably not true. But I think more likely in general, but there will be condensates that have the opposite effect.
Thank you, Julie. I don't know, Liz, if
So
you
yeah,
had some
I think I will. I don't have well, a few words just to close. I think this was an amazing webinar. Thank you so much for bringing us all together, Giuseppe. I think
My pleasure.
the scope of Allostery here is fantastic, and it's kind of given us marching orders for what to do next and inspiration. So thank you all for the time and for the discussion. It's been, yeah, wonderful. Thank you.
I really enjoyed all the talks. They were fantastic, both in terms of science and delivery. Thank you again. Really appreciate it. And thank you,
Beautiful.
Liz, for the opportunity.
Oh, thank you.
Thanks all.
Bye-bye.
Thank you. Bye-bye.
Bye-bye.